So we are telling about the pathogenicity and its virulence factor. So first virulence factor we can tell in this chapter is coagulase and other surface protein. So coagulase is very important. Staphylococci genus can be classified in as coagulase positive and coagulase negative organism. So Staphylococcus aureus is a very good example for coagulase positive organism. So first one is coagulase reacting factor, which is seen in plasma. So this is a free coagulase. Coagulase reacting factor is a free coagulase which is released by the organism into our bloodstream after attacking us or after attacking a patient. What happens is these Staphylococcus aureus release certain proteins which are called coagulase reacting factors and which can be detected in plasma, okay, which we can detect in plasma. Then next one is clumping factor or bond coagulase. This clumping factor is again a coagulase which is seen on the outer surface of the organism. So it is surface bond or bond coagulase. The first one is free and the second one is bond coagulase, okay, bond coagulase which is seen in the surface only, okay. Then next one is fibrinogen bind. Then how it works, okay, it binds to the fibrinogen, convert it to insoluble fibrin. Fibrinogen is a plasma protein which helps in clotting, okay. So it what happens here is this fibrinogen converts into an insoluble, okay, insoluble fibrin and clumping and aggregates happen. So the clotting will be affected by this. Then this is a primary test in identification of test coagulase aureus, sorry, staphylococcus aureus. So there are so much of biochemical tests we have to learn. So I will put videos on that too in the coming episodes. So you have to check that too. So for the coagulase test, so in the coming video I will be uploading the detailed video on coagulase test. And there are another other proteins too, or other surface proteins too, that is collagen binding protein, elastin binding protein, and fibrinolactin binding protein. These are all helps in escaping the immunity by the organism. Then moving on, staphylococcal enzymes. Okay, there are so much of enzymes produced by this. So first one is enzyme is a coagulase. That means it triggers blood clotting. Okay, it triggers blood clotting because this fibrinogen is converted into an insoluble fibrin, means that it causes a clotting. Then hyaluronidase, this hyaluronic acid, it breaks down the, this is an enzyme, ASC means it's an enzyme, okay. Here also coagulase ASE, okay. Here also ASE, that means these are all enzymes. And this hyaluronase, what they do is they break down the hyaluronic acid, okay, hyaluronic acid, and enables bacteria to spread in between the cells. So our body containing hyaluronic acid, and this enzyme produced by the organism breaks down this acid and enables the bacteria to spread from between cells. There in between two cells there is hyaluronic acid, and this enzyme will come and break down this acid, and what happens, the infection will spread from one cell to another. Then streptokinase enzyme, this what they do is this dissolves fibrin threads in the blood clot, okay. This breaks or dissolve fibrin threads in blood clots and allow it free itself from blood clots, okay. So coagulase causes blood clotting, and here what happens is streptokinase which dissolves the fibrin threads and the blood clots and free. Just imagine if the bacteria enters in an injured area, okay, there is blood in there, After some time the blood will clot. Maybe what happens is the bacteria is trapped in the clot. So at that time the bacteria produce staphylococcus and releases what the organism into our bloodstream or inside our body. Then there are some staphylococcal enzyme, that is extracellular enzyme. Coagulase is there, catalase is there, hyaluronase we already told, fibrinolysin is there, lipase is there, nuclease is there, penicillinase is there, beta-lactamase is there. Penicillinase and beta-lactamase act on the antibiotic resistance. Okay. Then fibrinolysin and hyaluronase helps in the spreading early. Okay. So moving on. So for a BSc person, this small description is more than enough for getting marks. Okay, for UG people, this slides, what are the points contained in this slide, these are enough. You don't need to do extra. Okay, then staphylococcus enzyme, so lipases is there which digest lipids. Okay, and allow them to grow on the skin surface in a cutaneous oil glands. Okay, and in cutaneous oil glands, so it helps in, this lipases helps in grow on skin by allowing what, lipases to digest lipids. Then beta lactamase, these break down penicillin. Okay, these break down penicillin and resist back beta lactam antibiotics to like penicillin and cephalosporin. So what they do is this break down penicillin. Okay, and resist another other antibiotics. Okay, so bacterial resistance occurs. Then moving on, then we are looking on the toxins of Staphylococcus aureus. It produces various toxins, alpha toxin, beta toxin, delta toxin, gamma toxin, pangent valentine toxin, exfoliated A and B, which causes exfoliated dermatitis or Staphylococcus scalded skin syndrome, that is SSSS. Food poisoning, performed toxin 2 to 6 hours, self-limiting disease. Food poisoning, it causes food poisoning. Again, it is self-limiting, 2 to 6 hours it will go away. Then super antigens are very important in case of staphylococci, that is enterotoxin 8, A to E and G to I. Then toxic shock syndrome toxin TSST1 is a super antigen again which activate a number of T cells. So for a BSc person, you have to study little bit detail about just few points about each toxin. So moving on. Um, then what we are telling is staphylococcus disease. So all the toxins are very important. So we cannot include detailed description on that toxins because this video will last long, so it will get lengthy. So what I'm doing is I'm going to detailedly tell each and describe each and every toxin, that super antigen, there will be one class about all these super antigens, so that will be uploaded in the coming episodes. So then what we are going to do, look for is the staphylococcus diseases. It causes various diseases, soft and skin and soft tissue infections like folliculitis, pharyngals, or it is called boils means water filled uh, like when a burn occurs there will be boils, right? Likewise pharyngals appears, abscesses, particularly breast abscesses occurs, wound infection, carbuncle, impetigo, paronychia, and often less often cellulitis too occurs. Then it causes musculoskeletal osteomyelitis, arthritis, then what pyomyositis, then respiratory infection like tonsils, pharyngitis, sinusitis, otitis, bronchopneumonia. Bronchopneumonia means our bronchus are affected and causes pneumonia. Lung abscesses, emphysema, rarely pneumonia. Then next, central nervous system is also affected by Staphylococcus aureus, which causes abscesses, meningitis, intercranial thrombophlebitis, then endovascular bacteremia, septicemia, pyemia, endocarditis, etc. Then urinary infection, uncommon, and UTI. Okay, and UTI, that is Staphylococcus saprophyticus, which cause females UTI in female, and which is called as honeymoon UTI, which is more affected in sexually active women. Then novobiocin resistant. Then these are some pictures of staphylococcus infections. Then moving on, then there is bacteriophage typing. Okay, bacteriophage typing is the international basic set of phages for typing staphylococcus human origin. Okay, group 1, group 2, group 3, group 4, group 5, and not located. There are certain strains too, 29, 52, 52A, 79, 80, 3A, 3C, 55, 71. So this is not that much important, but you have to keep it in mind. There are there is bacteriophage typing, and there are certain groups, group 1 to 5, and there are certain strains too. Then epidemiology, primary parasites of humans. Okay, it colonizes in the skin glands, mucous membrane. Human patients and carriers are there. Potent source of this is a very potent source of infection. Patients and carriers. Carriers are those persons who harbors the organism even after they get cured from the disease. Potent source of infection. Animals and inanimate object, less important. Okay, animals and inanimate objects, like not that much important but less important. 10 to 30% healthy population has staphylococcus as its nasal carriers. So if we test a nasal swab, we can there is chances of getting what, in 100%, 10 to 30% will have this staphylococcus carry staphylococcus organism. 10% perineal carriers, 10% hair carriers, 5 to 10% vaginal carriers. During menses, tampons, those women which uses tampons during their menses are likely to cause toxic shock syndrome. Moving on, Staphylococcus carrier stage. It starts early in life. Colonization of umbilical cord in neonates. Even what happens? This Staphylococcus can get colonized in the umbilical cord of neonates. Shedders. Shedders means what? These are carriers disseminate large number of organisms for a prolonged period. Shedders means these uh, people or these carriers shed this organism for a prolonged period. Okay, prolonged period. Then cocci shed patients and contaminated formats like kitchen handkerchiefs, bed linen, blanket persist for days and weeks. Mode of infection. Exogenous infection is there and endogenous infection is there. Exogenous means it causes outside. Okay, and endogenous means these are infection which get into our body. Mode of transmission. Direct contact is there. Indirect contact by formalities is there. Dust, droplet nuclear infection, etc. Then moving on, nosocomial infection. This is very important. When a person is admitted into a hospital and he is getting a disease from the hospital itself or hospital surrounding itself, then it is called nosocomial infection. Healthcare and hospital acquired infection, multi-drug resistant bugs are there. So MRSA, very very important. I'll give another video on this. Methicillin resistant staphylococcus, that is alteration of penicillin binding protein 222A. Mutation of MEC gene is there. Short arm of chromosome 6, CA-MRA, MRSA 20%, HA-MRSA 80%, E-MRSA. So you just note all these points. I will give a detailed video on all these things. These are the characteristics of CA and HA-MRSA. CA is a what community acquired one, and HA means hospital acquired. CA means community acquired MRSA. HA means hospital acquired. Community acquired is only 20%. Hospital acquired is more, which is 80%. This affects community acquired mainly affect young and healthy persons with no recent exposure to healthcare, while this previous contact with any healthcare setting like hospital, nursing homes, or hemodialysis centers, etc. Type of infection means that this is mild to moderate severity, causing skin soft tissue infection are more common. And hospital acquired is more severe invasive disease, hospitalized patient or frequent contact with healthcare persons. Skin and soft tissue and lungs are affected. Here, bloodstream, lungs, surgical site, prosthetic implant, etc. So this is a calculation of USA that we don't need to study. So what are the risk factors of MRSA infection? The injection, drug abusers, skin trauma, then higher body mass index, cosmetic body shaving, physical contact with the person with the draining lesion, incarceration, previous skin infection with MRSA, homosexuals, military recruitment, and mental asylum. So moving on, lab diagnosis, samples to be collected and transported to the storage. It is common for most of the all bacteria. So and there are certain BPA blood agar, McConkey, nutrient agar, selective mediums like Lutlam. Nutrient agar rich could produce golden yellow pigment. Biochemical tests include catalase, gram staining, tube coagulase, mannitol fermentation, phosphatase, then indole, MRVP, urine hydrolysis. All these tests I will include different types of different videos for all these tests individually. Then this is the gram staining. Here you can see the single celled organism clumped together. I know this picture is not that much clear. So for nasal carriers, what we do is we take nasal swab and culture it, and this is the cefoxitin screen for MRSA. If it is sensitive, that means these are MRSA strains. Then treatment for MRSA is hand washing, chlorohexidine spray, muco, uh, mupirocin topical, vancomycin, linezolid, taucoplanin, and these are cephalosporin, fifth generation cephalosporins can be used. Tropical application we can use Bactroban. Then Staphylococcus aureus antibiotic susceptibility test we can use. MRSA is resistance to this penicillin, ampicillin, amoxicillin, cloxacillin, cefixone, cefixtidin. So a good marker for MRSA. We use Kerber disk diffusion method, MIC E-strip method, or we take a novobiocin sensitivity. Then bacteristatin resistance, fluorescinoid sensitivity can be used to screen Staphylococcus aureus. Okay, so there is so much of common things, so you have to prepare note and study for your exams. So thank you. Today's video is this much. I will give detailed explanation for this super antigens and biochemical test in the coming videos. So thank you so much for watching. If you like the video, like and share to your friends if it is useful, if you think it is useful. And so that's it, guys. Like, share and subscribe my channel to get more videos. Press the I button to get continuous videos. Thank you.